Hello, everyone. My name is Amanda Grimm. I'm the Senior Segment Marketing Manager here at GenScript for our IVD portfolio. My presentation today is called From Insight to Impact, Enabling Sensitive, Specific Point-of-Care Assays for Infectious Disease. Contents of this presentation will include some information about GenScript. I'll go into an overview of infectious disease. We'll go through some of GenScript's solutions. I have some customer highlights and GenScript applications to share with you, and then we'll close out with a summary. Let's start with some information about GenScript. GenScript has over 20 years development experience, having initially started out in the gene synthesis space. They do continue to innovate and upgrade current technologies for their services and products. And they do also have regulatory experience due to the navigation of diagnostics during the COVID-19 pandemic. Looking at the history and milestones of the GenScript group, GenScript was founded in 2002 in Piscataway, New Jersey, and since then has expanded the umbrella to include BestSime, Legend Biotech, and ProBio. Here we'll go ahead and just take a look at the subsidiaries a little bit closer. GenScript, the life science CRO, that is the group that I am a part of. We have ProBio, which is our biologics CDMO, Legend Biotech, responsible for cell therapy developments, and then we have BestSime, conducting synthetic biology. Go ahead and move into the overview. We'll start with the global reach and scale of infectious diseases. Infectious diseases affect hundreds of millions globally, with over 40 countries in 2024 that face severe outbreaks. The factors behind these resurgences include declining vaccinations, reduced immunity, and climate change that contributed to the increase in these outbreaks. We also had a dengue fever surge. Argentina experienced a dramatic 151-fold increase, exceeding 488,000 reported infections. So there is a need for global response related to enhanced health monitoring, vaccination campaigns, and internal cooperation in order to mitigate the impact that these diseases can have. This slide is a look at the most prevalent and rapidly spreading infectious diseases according to WHO and other global surveillance data. It looks at the countries as well affected by each disease. So the diseases that are most prevalent include dengue fever, measles, whooping cough, tuberculosis, polio, monkeypox, cholera, and influenza and RSV. Slide is a chart compiled of diagnostics that are used for some of these infectious disease diagnoses in order to make sure that individuals who may be infected can be diagnosed sooner, getting them treatment sooner, improving the opportunity for recovery. So we'll look at a couple of these here, the top one being dengue fever, the diagnostic method being the NS1 antigen test or IgM antibody test. There's also nucleic acid amplification tests like RT-PCR or RT-PCR. These are antigen or antibody tests, or also nucleic acid. The use case for this specifically would be for early detection in that 0 to 7 days post-symptom onset. A means of utilizing this would be for outbreak control. Limitations with this type of test do include cross-reactivity with other flaviviruses, limited sensitivity in those later disease stages. Another one we can look at is influenza down there towards the bottom. We have lateral flow for viral antigens, nucleic acid amplification tests for RNA. These are used for rapid diagnosis in emergency and outpatient settings. The limitations of these type of tests include lower sensitivity of antigen and also some seasonal variability. So here we can take a little bit of time to look more in depth at some of these different formats that were mentioned in that previous slide. We have the lateral flow assays or rapid tests, where the principle of these is essentially immunochromatographic technique using capillary action to detect antigens or antibodies. These can be used for diseases like COVID-19. We saw that listed for influenza and dengue. It can be used to multiplex as well. So this can be done and achieved by placing multiple capture zones or spots on a single strip, or sometimes we see some of these, they're kind of next to each other in the same kind of cassette. The advantages of these types of tests are that they're low cost, they're portable, they're very easy to use. The limitations with these do tend to be the lower sensitivity. Sometimes we see cross-reactivity, also high antibody consumption. Another format is LAMP, or loop-mediated isothermal amplification. The principle here being the DNA amplification can be done at a constant temperature using specifically designed primers. So this is important because it often removes the need for the thermocyclers used for PCR. These can be used to detect bacterial, viral, or parasitic DNA, RNA. These also can be multiplexed, although it's complex due to the primer design. Advantages include that it goes back to the fast and sensitive, being fast and sensitive, similar to PCR, really highlighting the removal of needing that thermocycler. The limitations do refer back to multiplex being difficult and the risk of non-specific amplification. The next format to look at would be nucleic acid amplification tests, specifically multiplexing in this format. This is going to allow you to simultaneously amplify and detect of multiple nucleic acid targets. So we can see this being used with syndromic panels for respiratory, gastrointestinal, or even STI pathogens. The formats for this include PCR, RT-PCR. More recently, we're seeing CRISPR involved with CRISPR Enhance, like with the Sherlock and detector formats. The advantages here being that high sensitivity and specificity, broad pathogen coverage, although the limitations do include cost, complexity, and equipment needs. Let's go ahead and move into, for those formats, the different biological and synthetic components that would be required to develop and manufacture. So we have antibodies and antigens. Antibodies can capture the specific antigens in immunoassays like the lateral flow and like we see in traditional ELISA to detect pathogens. We have CRISPR proteins and guide RNAs. The CRISPR proteins Cas12a and 13a do detect nucleic acids with very high sensitivity, guided by that custom gRNA target. We have synthetic DNA and aptamers, where synthetic DNA templates and aptamers provide precise target recognition and that signal amplification in these assays. And then we have reporter molecules. These emit the signals through cleavage during detection, enabling measurable readout in these diagnostic tests. Having looked through some of the prevalent infectious diseases and some of the different diagnostics and point of care involved in diagnosing patients, We'll take a look at the challenges in point of care development specific to some of the formats I referenced in the previous slide. Um, These challenges include sensitivity and specificity. These point of care require balancing that rapid detection with precise sensitivity and specificity to avoid misdiagnosis. We have reproducibility challenges. The consistent performance is challenged by the variability in batches, in users, and environmental factors like temperature. And then we have scaling and manufacturing. The production of these devices requires quality control, a stable supply chain, as well as regulatory compliance. And going into the next section, we'll look at ways that GenScript and the products and services for the IBD portfolio can help address these different challenges in your development. Okay, let's go ahead and start with GenScript solutions. So here we'll go ahead and start with the development workflow. This really starts with that disease of interest. So whether you're working on dengue fever or influenza, you'll want to identify which target your diagnostic is going to be looking for, whether that's a protein, antibody, or the nucleic acid. Your diagnostic would then move into design and optimization. So you're going to be testing the format that you've chosen, whether that's next-gen sequencing as an example, ELISA, PCR. And then the test would move to validate and verify. And then once that data is collected, reviewed, and submitted, the approval process would then allow you to utilize it as a diagnostic. Related to that development workflow, GenScript identifies three main stages for assay-specific solutions, that being the target identification stage, a number of resources for gene and protein analysis, and then once you move into your format and validation stage, we have different solutions for antibody antigen detection, and then another for molecular detection. Let's go ahead and dive into the first challenge with sensitivity and specificity. I think this can really take shape through the target characterization, because here you're going to be identifying unique biomarkers like antigens and the host immune response in order to enable that accurate point of care development. We can also further look at the disease pathway to better understand this, like the infection timeline. This is going to guide that optimal sample timing as well as target selection for tests. And then being able to develop on that the test sensitivity and specificity. So accurate knowledge ensures tests detect disease effectively across all of the progression stages. And then the risk without understanding lacking that foundational knowledge will ultimately lead to ineffective or misleading point of care results, especially in complex cases. To support that characterization of targets, our Gene Titan platform can really support evaluating target genes and their functionality. It's a proprietary arrayed semiconductor-based electrochemical oligonucleotide synthesis technology that allows precise synthesis of each individual oligo synthesized on a chip. This technology can also be combined with either hybridization or amplicon-based enrichments for next-gen sequencing, and then it can also be used to create customized guide RNA libraries that could be cloned into either dual or all-in-one CRISPR vectors for additional methods to study gene functionality. We do have ready-to-use guide RNA libraries for commonly investigated diseases like Alzheimer's or diabetes, or we can customize specific to your area of study as well. I do want to share a case study with you specific to using the GeneTitan technology for a customized CRISPR gRNA library. Here we have a customized library that contains over 58,000 unique guide RNAs for gene knockout in the human genome. All of these guide RNAs were synthesized simultaneously as short single-stranded oligonucleotide using our arrayed chip platform. The conserved sequences were added to the end of the guide RNA to facilitate PCR amplification. Following synthesis, these guide RNAs were amplified to double-stranded PCR products and cloned into the lentiviral vectors to create the library. Overall, the quality of the library was determined by assessing library diversity and sequence distribution using next-gen sequencing. 30 clones were randomly picked and sequenced using Sanger sequencing. The sequencing results showed that all of the sequences were unique with 100% diversity among the randomly picked clones. Additionally, each guide RNA sequence exactly matched a desired guide RNA sequence, indicating accurate oligonucleotide synthesis and successful PCR amplification of the guide RNAs following that synthesis. CRISPR nucleases play a critical role in gene editing and can be added to create a model to better understand the intricacies of disease and the potential for protein-protein binding. We do offer a variety of off-the-shelf nucleases such as Cas9, 12A, or 13A, as well as prime editors in order to customize your gene editing strategy with that incorporation of our single guide RNA service. This allows you to create a couple of targeted edits, or you can add to your custom library to enable high throughput screening. For protein expression, whether your antigen is simple or complex, we have a range of platforms to find your best fit. The E. coli platform is great for antigens that are relatively low maintenance. Our insect platform helps with soluble, membrane, or more complex proteins. Our mammalian platform supports various antibody or recombinant proteins that need to be closer to biologically active. And then we recently launched our cell-free platform. This is great if you're doing computational or AI design. This is a great option too to be able to test hundreds or even thousands in a high throughput format. This does also include a kit version, which I'll talk about more in the next slide. The new CF Express kit launched just a couple weeks ago, and we've received really positive feedback so far. I want to share some additional details on the kit since it is discounted until the end of September. It is an E. coli extract-based kit that will synthesize proteins in just a few hours. It includes the cell-free extract, protein synthesis buffer, and a control plasmid. The protein synthesis capabilities cover a range of protein types with high yield results, and it's going to be a good option for proteins that may be toxic to a cell system. To add to the protein service, we also have a protein analytical service in order to enable that antigen creation and protein interaction studies. This is an advanced integrated system designed for in-depth study and evaluation of protein characteristics. It offers a wide range of services, including qualitative and quantitative analysis, protein modification analysis, impurity analysis, molecular interaction, and more customized analysis. I have a case study here showing that protein analytical integration and what it might look like with the utilization of the TurboCho mammalian protein expression platform that we have. This is actually a scalable platform that I was going to talk about later in the presentation, but I do want to show how this can be applied and integrated. So in these earlier stages with the high throughput and the expression, early in the development stages, you could add qualitative and quantitative analysis that would include the purity by SDS or HPLC. We have affinity screening, ranking, as well as some of those more customized services available. As you scale up into the later stages with assay validation and optimization, there's the option with protein modification and molecular interaction analyses, where you can look at identification by mass, post-translational modifications, glycan analysis, binding affinity, and more. Next, we offer a range of options for antibody development, from polyclonal to traditional mouse hybridomas to our positive cell enrichment platforms. Each platform is customizable based on your antigen and immunization choices. The positive cell enrichments include our monorab, rabbit monoclonal, and our newer turbomab monoclonal options. Using rabbit as a host provides unique advantages due to their production of highly sensitive and specific antibodies that are going to have little to no cross-reactivity. The turbomab platform is an advanced hybridoma platform that is going to enrich the positive cells before fusion in order to increase the likelihood of specific antibodies. Both of these can be expedited as well by adding the B cell screening option. Showcase the monorab platform, I have a case study here to share where we looked at anti-beta-actin, monoclonal antibody produced for IHC. We generated antibody in both mouse and rabbit, and this data can be seen in figure 1, where picture A is the mouse anti-beta-actin and B is the rabbit. In C, this significantly shows more binding or better affinity of the rabbit anti-beta-actin as highlighted by the white arrows. And then in figure 2, we compared the anti-rabbit beta-actin to 2 competitors and found ours to have higher sensitivity compared to competitor A and higher specificity than competitor B. An add-on to your antibody development project could include conjugation and labeling. For antibodies, we have biotin, horseradish peroxidase, or fluorescence. By binding the fluorescence in a direct format, this can help expedite the staining protocol, as well as reducing the background and also simplifying multi-signal assays. We do also have the option and capability with oligo conjugation. Ultimately, this would be an oligostrand or a DNA molecule that can be conjugated to an antibody, a peptide, or a protein. And applications for this could include immuno-PCR, an immune assay, or a proximity ligation assay. Some other antibody add-ons may include our specialized antibody services. We have a number of single-chain variant fragment generation packages, as well as antibody pairing packages. We can also perform independent antibody validation. We can also assist with antibody optimization through our DNA mutant libraries. These are common with antibodies if you're looking at the hinge engineering, the CDR and variable region engineering, or even the FC region engineering. Let's move into the second challenge of reproducibility and what GenScript offers as solutions. As you've moved into your assay design selection, you want to limit a lot of variability with your raw materials. And as you identify controls and run conditions, ensure that you have reliable and well-characterized materials. We have a pretty comprehensive portfolio of off-the-shelf IVD antibodies. These have complete characterization, identified pairs for common targets across the most commonly investigated diseases like diabetes, Alzheimer's, and some infectious diseases like HIV and HCV. Additionally, they have superior lot-to-lot consistency and follow a highest standard quality control from the National Institute for Biologics. Our oligosynthesis service offers highly precise and sensitive custom diagnostic probes and primers. You can expect low exogenous contamination, enhanced fluorescence, and stable quality. To demonstrate the strict control of exogenous contamination, I have this case study I can share for qPCR oligosynthesis. The data on the left uses FAM and NED fluoros, and we can see the tight overlays for each red and blue curves confirming the batch-to-batch consistency. The same can be seen for the data on the right using targets with FAM and VIC fluoros. Both of these achieved high precision with low CT values for high sensitive outcomes with high detection rates. We have a number of off-the-shelf products to support enhancing reproducibility. This includes a range of PCR reagents like Taq polymerase and the nucleotides. We also have different protein purification options like resins or magnetic beads. And then we have instrumentation that can help to automate your workflow, like our newly launched AMAG Quattro ProAb 1300. This is a fully automated instrument that uses magnetic beads for the purification of proteins or antibodies. Let's move to our last challenge for point of care development, that being scale up. So GenScript does ensure your prototype materials seamlessly transition to large scale production, preserving the sensitivity, specificity, and reproducibility. Our technologies optimize that cost efficiency and consistency, empowering you to deliver high performance point of care at scale. The first of these being our TurboCho platform. It has been mentioned already in a couple places leading up till now, but this is a scalable recombinant protein expression platform that's going to move with you through the development process from early discovery with transient high throughput to express, and then to later stage stable expression with high performance and stable expression options. Our team recently upgraded the high throughput and express to improve both the yield and speed. We can also scale antibody yields through our large-scale antibody production service. This slide here demonstrates our ascites production, and we can deliver 2 to 5 mils of 1 to 10 mg per mil of monoclonal antibody. And then alternatively, we can also scale up hybridoma or recombinant antibody production. For hybridoma, we can deliver 15 to 50 mg per liter, and for recombinant antibody, over 100 milligrams per liter. In this next section, I want to share some customer highlights to show you how they are using some of the services I shared today for their diagnostic research. And then I wanted to share the different COVID diagnostics that GenScript commercialized during the pandemic, utilizing services and products that we offer. For the first customer highlight, I have this group looking at the establishment of a CRISPR-Cas9 knockout library for screening type 1 interferon-inducible antiviral effectors in pig cells. And so here the goal was to construct a CRISPR-Cas9 knockout library of over 1,800 unique sequences targeting 359 ISGs with an average coverage of 5 to 6 guide RNAs per gene. The outcome was that this ISG targeting CRISPR-Cas9 knockout library did demonstrate a high level of consistency between type 1 interferons, including 2 subtypes of interferon A and 1 subtype of B, and at a high rate of hit confirmation, demonstrating the promise of wide application in screening novel sets against a panel of porcine viruses. The GenScript service and products used in this study was our Gene Titan semiconductor technology in order to generate the single guide RNA library. And then we have our ready-to-use interferon alpha 1b and 2a and the interferon beta cytokines. For this second customer highlight, we have a group looking at the analysis of whole genome to identify Alternaria species. And the goal in their study was to develop a robust and efficient method to identify these Alternaria species using the analysis of the whole genome. The method aimed to facilitate the timely detection of these species, enabling targeted treatments and mitigating associated changes. The outcome was that the study was successful to construct a species-specific target sequence library for 31 Alternaria species. The method was validated using Sanger sequencing and CRISPR-Cas12a technology in order to achieve accurate and rapid identification of the target species. The GenScript service used in this study included GenScript generation synthesis of crRNAs for the CRISPR-Cas12a system. These were crucial for the rapid and precise identification of genomic DNA samples, and these were synthesized to target species-specific sequences that then enabled the detection of the Alternaria species with high specificity and sensitivity. So next I want to move to share the GenScript diagnostics, and just to preface that many of these products are no longer available. The purpose here is really to share our development and commercialization achievements. The first one here on the screen is our CPAS SARS-CoV-2 neutralizing antibody detection kit. The clinical indications for this kit was to detect anti-SARS-CoV-2 neutralizing antibody in patient serum and plasma. This ELISA did achieve multiple regulatory approvals in multiple countries around the world. It did include an RUO version for research and testing outside what it was initially approved for. One of those examples was for some research with wild deer exposure surveillance. The technology and kit was commercialized from the work done by Dr. Lin-Fa Wong with Duke NUS. The technology for this ELISA was essentially the replication of conventional virus neutralization testing into a surrogate technology that allowed for a high throughput testing format and then eliminated the need for advanced biosafety conditions. When we look at the mechanism of SARS-CoV-2, the spike protein of the virus would bind our human ACE2 receptor. And so by our body producing these neutralizing antibodies, these would then bind the RBD of the spike protein, preventing the virus from binding to the receptor. What we did was convert that into an ELISA format using a recombinant ACE2 receptor protein bound to the ELISA plate, using the RBD, conjugating that to HRP, so that if that was able to bind to the plate, that a color developed. So if the patient had neutralizing antibody, this would bind the RBD and it would be washed away. So it's a little bit of a reverse concept where negative samples generated a color, but if the patient had neutralizing antibody, no color, no or little, very little color would develop. The overall components for this included that antigen-coated plate with the ACE2 protein, positive negative controls, the recombinant RBD HRP conjugate, some different solutions, buffers, and a wash solution. So somewhere around mid-pandemic, that same technology from the ELISA was converted into a lateral flow rapid test format. This did receive the CE mark in the EU, specifically the CE 1434 for direct-to-consumer sales. To expand a little bit more on the technology and how it was applied into lateral flow. Ultimately, the principle of this was the lateral flow latex immunoassay and used immunochromatography blocking sandwich method in order to detect the SARS-CoV-2 neutralizing antibodies in the sample. So the conjugation pad contained RBD and chicken IgY labeled with latex microspheres. The nitrocellulose membrane was coated with ACE2, the blocking line, and then we had RBD as the test line and the anti-chicken IgY antibody as the control line. If the sample contained neutralizing antibody, a red line appeared on the test pad or the T line, and the red line would then be read as a positive result. The neutralizing antibody content is positively correlated with the depth of color of the T line and could be assessed using a control color card that was provided in the kit. If the sample did not contain neutralizing antibody, then no red line developed. In the pandemic, GenScript did have a qPCR detection assay where the clinical indications for this was to detect SARS-CoV-2 nucleic acid in patients' nasopharyngeal samples. This did receive the CE mark in Europe. It was RUO in the US, but overall it was a TACMAN qPCR assay that targeted the ORF1 AB, the RDRP, N, and E genes in the SARS-CoV-2 genome. So just a little bit more information related to the commercialization of the qPCR kit. The development of this was based on early and available coronavirus research at the time. Customers could choose to do single or dual labeled testing. And the output was quantitative measurement using qPCR instrumentation. And that brings us to our last section, where I'll go ahead and summarize the information that was presented today. We'll just take a look back at the development workflow when it comes to identifying the disease of interest, moving to identify the target, design and optimize the test format, validate and verify, and then upon approval, use it for diagnostic purposes. GenScript separates the development workflow into 3 stages, that being target identification, assay format and validation, and assay production and quality control. Once you've identified the format, the solutions become more assay specific, whether you're looking at antibody antigen detection or molecular detection. The challenges that I addressed today when it comes to point of care test development included sensitivity and specificity, reproducibility challenges, as well as scaling and manufacturing. And those can be addressed at the different stages in the development workflow, target discovery and characterization, really helping you to fully identify your target types, confirm disease pathways, determining that cause and effect, and evaluating protein-protein interaction. Once you've decided on your assay design selection, making sure that you have consistent lot-to-lot consistency, as well as fully characterized raw materials, supporting you through your control identification and run conditions, as well as the optimization of your assay. Lastly, when it comes to assay production and quality control, making sure that you have the capabilities to scale up in order to run field and clinical testing, your raw material lot production, validating via regulatory processes, and then to be able to implement the quality control processes. By sharing with you a couple of customer highlights, this also puts the spotlight on our customer success and over 20 years of partnership that we have had supporting them. You can see here that we've had more than 100,000 successful customer research publications in peer-reviewed journals. That includes the Cell, Nature, and PNAS. We are continuing on an upward trend of citations in our customers' publications for our products and services, and we look forward to continuing that growth and those partnerships. And that concludes my presentation today. I thank you for your time. If you have questions, please reach out to us. I encourage you also to visit our virtual booth. We have a number of different resources and brochures and flyers related to a lot of the products and services that I shared with you today. We also have our chat feature enabled to be able to answer any questions that you may have. So thank you.